And, and my name is Kohji Fukunaga from Tohoku University. I'm also CEO in BRI Pharma. Thank you for your kind invitation in this conference. Today, I talk that the multiple sclerosis induced mitochondrial injury is mediated by fatty acid binding protein 5 and 7 in the oligodendrocyte. In multiple sclerosis, the autoreactive lymphocyte attack myelin that covers nerve fiber and causes communication problem between the central nervous system and the peripheral muscle and organs. The symptoms affect movement disorder, vision problem, speech, pain, and bladder functions. Multiple sclerosis is caused by brain inflammation after blood-brain barrier disruption. BBD disruption causes autoreactive lymphocyte activation. The lymphocyte activation next causes activation astrocyte microglia, thereby releasing the cytokine, interleukin-1 beta and TNF-alpha. Fatty acid bioprotein, which is a transporter of fatty acid in the brain. Recently, we discovered that FABP5 accumulation to mitochondria promote VDAC1 oligomer formation, which trigger release of the mitochondrial DNA and cytochrome C, thereby inducing the apoptosis and oxidative induced cell death in oligodendrocyte. So, we developed the FABP5 and 7 inhibitor to prevent the multiple sclerosis symptom in mice. Cyclosane and arachidonic acid promote radical oxide species production in oligodendrocyte, thereby causing synuclein oligomer formation through the fatty acid binding protein 7 and mitochondrial damage through FABP5. As shown in this figure, FABP7 make a complex with synuclein, making the oligomer of alpha-synuclein, and FABP5 is accumulated in mitochondria under the oxidative stress, thereby killing the oligodendrocyte. We have been developed FABP inhibitors. So among the FABP inhibitors, we selected ligand 6 named BRI 601 because this ligand has high affinity for FABP7. We also selected ligand 8 because this ligand has high affinity for both FABP5 and 7. Previously, we reported that cyclosane oxidative stressor make oligomer formation of alpha-synuclein in oligodendrocyte. Treatment with BRI 601 inhibits the oligomer formation of alpha-synuclein. Likewise, cyclosane treatment causes oligomer formation of FABP7, which is also inhibited by treatment with BRI 601. We next focus on FABP5 under the cyclosane exposure. As shown in left side, after exposure cyclosane, FABP5 is accumulated in mitochondrial fraction. As shown in the right side, FABP5 showing the red accumulating in the mitochondrial fraction. This accumulation causes the fission of the mitochondria and elevation oxidative stress in the mitochondria. In this slide, we also show that FABP5 is accumulated in the oligodendrocyte after exposure to lisophosphatidylcholine, which is also toxic to the oligodendrocyte. The formation VDAC1 oligomer partly inhibited by treatment with BRI 601 in KG1C cells. In this slide, we define the effect of the FABP5, function of FABP5 in VDAC1 formation. As shown right side, exposure to cyclosane make oligomer formation of VDAC. This VDAC oligomer formation inhibited by treatment with FABP5 shRNA to knock down FABP5, suggesting that FABP5 trigger the formation of the VDAC formation, VDAC oligomer formation in mitochondrial membrane. To support the effect of the FABP5 sub-inhibitor, we choose the EAE mouse model. EAE mouse model was induced by myelin peptide and pertusstoxin. BRI 601 administered by orally after onset of EAE. The treatment improves the motor deficit in EAE model. We next measure the activation of the astrocyte and microglia in the EAE mouse. You can see the activation astrocyte and microglia measured by caveolin and EVA1 completely inhibited by treatment of BRI 601. The production cytokine such as TNF-alpha and iron beta are also inhibited by BR 601 administration. On the other hand, expression myelin basic related protein is recovered by treatment with BR 601. Our compound suppresses the progression of the multiple sclerosis, we should define the early phase patient in multiple sclerosis. To do so, we develop biomarker to detect brain inflammation using the FABP3 and the FABP5. We could distinguish the dementia patient from the healthy control. As shown left side, using the FABP3, the level of the serum, level of the FABP3 in the plasma are upregulated in dementia patient, including Alzheimer, MCI, and Parkinson, and DLB. Using this system, we could detect the brain inflammation in the multiple sclerosis patient, so that this diagnostic technology is useful for the clinical evaluation of our compound. So, this is a take-home message. Mitochondrial impairment has been documented in the brain and spinal cord in multiple sclerosis patient. Today, I confirmed that FABP5 is accumulated in the mitochondria in EA model and promote VDAC4 formation, which trigger release of cytochrome C and mitochondrial DNA from mitochondria. FABP5 and 7 inhibitor, such as BRI 601, prevent the release of the cytochrome C and mitochondrial DNA from mitochondria, thereby inhibiting progression of the EAE syndrome. The biomarker detecting brain inflammation are also developed in BRI Pharma. We are now looking for the collaborative company to promote the translational study of BRI 601, targeting mitochondrial protection in multiple sclerosis therapy. Please contact us to promote our collaboration. Thank you for your attention.